Welcome to the deep dive. Today we're taking a closer look at a really groundbreaking genomic technology, optical genome mapping, or OGM. As clinicians and researchers, we all know that interpreting the human genome, well, it isn't just about reading the A's, T's, C's, and G's. It's crucially about understanding structural variations, SVs. Those large-scale rearrangements, insertions, deletions, they often drive genetic disorders and pathogenic conditions. And traditional methods, like short read sequencing, they frequently just miss these critical long-range changes.
Yeah, what's truly revolutionary here, I think, is OGM's ability to directly visualize and analyze these ultra-high molecular weight DNA molecules,
Yeah.
really long strands. This gives us an unparalleled genome-wide assessment of structural anomalies that were, frankly, very challenging to characterize before, often needing multiple tests, costly, time-consuming.
Exactly. So our mission today is to really unpack OGM. We'll explore its advanced capabilities, how it precisely resolves some incredibly complex genetic puzzles, and its rapidly expanding utility in clinical diagnostics, from constitutional disorders right through to acute leukemias. We'll delve into some recent research that showcases OGM's power, highlighting why it's becoming such an indispensable tool in the modern genetic laboratory. Okay, let's start with the foundations then. What exactly is OGM and what gives it this distinct advantage in visualizing the genome compared to, well, what we're used to?
Well, OGM uniquely analyzes these ultra-molecular weight DNA molecules, UHMW DNA. Typically, we're talking over 300 kilobase pairs long. And this scale is vital because it lets OGM capture that long-range genomic information, precisely what's often missed by short-read sequencing because of, well, its inherent limitations with read length.
So how does OGM actually see these extremely long molecules? It's not base-by-base sequencing, right?
Correct. It's quite different. The process involves fluorescently labeling the DNA, but only at specific sequence motifs. A common example is the CTK hexamer sequence. This creates a distinct sequence-specific pattern of fluorescent signals. Think of it like a barcode. You get roughly 14 to 17 signals per 100 kilobase pairs across the whole genome. Then these labeled DNA molecules are carefully linearized, basically stretched out, by flowing them through tiny nanochannels. And while they're in there, they're imaged and their fluorescent patterns are digitized for computational analysis. Comparing the patterns tells us about the structure.
That's a really unique approach. What kind of resolution are we talking about for structural variants? And crucially, what are the practical implications? I'm thinking cost, throughput.
Good question. OGM can detect structural variants down to about 500 base pairs in size. And here's where it gets really compelling clinically.
Yeah.
The throughput is high. You can get about 200x genomic coverage for approximately $500 per sample. Now compare that to long-read whole genome sequencing, which, as you know, can range anywhere from $10,000 to $20,000. So it's a substantial cost advantage, making comprehensive SV analysis much more accessible.
Okay, that cost efficiency in the long-range view, that's incredibly powerful. But, in the nuanced world of genomics, standard often isn't enough, is it? What happens when we need even more precision? Especially in those tricky genomic regions or maybe for specific targets. This seems to be where multicolor mapping comes in, like the work by Upaluri and colleagues.
Exactly. And this naturally leads us to ask, well, how do we achieve truly site-specific labeling? How do we overcome the known limitations of just relying on those standard motifs? Because motif-based methods, while they're robust generally, they can struggle where motifs are unevenly distributed or in highly repetitive regions. That makes precise breakpoint localization difficult sometimes.
Right, I see. So how did Upaluri et al. Tackle these limitations? What's the core idea behind their multicolor strategy?
It's quite clever. They combine the conventional sequence motif labeling with a Cas9-mediated target-specific labeling approach. So two layers of information. They use a direct label enzyme, DLE1, to put, say, green fluorophores on the common motifs like TTT-AG, standard OGM backbone. But then for that finer site-specific targeting, they use Cas9. And this lets them target basically any 20 base sequence, a 20-fomer, and label that with, say, red fluorophores.
Ah, so you get two distinct color signals, almost like a dual color barcode system layered on top of each other, giving you a richer data set, more precise information.
Precisely. This combined approach still allows for robust SV detection, but critically, it lets you use custom labels to interrogate genomic features that aren't accessible just by motif labeling alone. And that's key for precisely locating breakpoints and, importantly, accurately estimating copy numbers of genomic repeats. That's a significant step up.
And how did they validate this? I mean, did they show it works in clinically relevant scenarios? What were some key examples?
Yes, they did several critical validations. One really standout example was quantifying D4Z4 copy numbers. As you know, this is a 3.3 kilobase pair tandem repeat on chromosome 4q35, a key biomarker for facioscapulohumeral muscular dystrophy, FSHD. Now, this region inherently lacks the common OGM motifs, making it hard for standard OGM. But their DLE-Cas9 method accurately estimated the copy numbers on both alleles 4QA and 4QB. The accuracy was impressive, less than a single copy variation. And that level of precision is absolutely crucial for a definitive FSHD diagnosis.
That's very specific. What about other structures? Did they look at telomeres, for instance?
They did. They successfully assayed telomeres, those TTA-GG repeats at chromosome ends, up to 20 kilobases long. These are vital clinical biomarkers for aging-related diseases and also some cancers. Their methods significantly improved on previous techniques. It allowed them to characterize telomeres even in regions that were previously unmappable, maybe due to fragile sites or just lack of suitable reference sequences nearby.
Interesting. And what about mobile genetic elements, things like line 1 insertions?
Yeah, they applied it there too.
Mm.
To discover transposable line 1 insertions across the genome. Line 1s are, everywhere, about 17% of our genome. And when they insert into genes, they're associated with various cancers, hemophilia, muscular dystrophy. The method successfully detected 55 line 1 insertion sites in a standard reference sample, NA12878. It picked up 51 out of 52 previously reported ones and found 4 new ones, so high sensitivity and discovery power.
So this multicolor approach, it sounds like it's not just more accurate and versatile, but maybe also easier to perform than earlier attempts at dual labeling?
Yes, that's another advantage they highlighted. Compared to a previous method using NT-BSPQI and Cas9, this new DLE-Cas9 approach seems to preserve DNA integrity better, so you get longer molecules for analysis, which is always good. The protocol itself is simpler, apparently with higher efficiency for incorporating that second red label. And it's all done in a single tube reaction. They even showed potential for multiplexing up to 200 different guide RNAs in earlier work, suggesting it could be scaled for high-throughput targeted analysis too.
Okay, so moving beyond just detecting these SVs, the ability to translate all that raw OGM data into a comprehensive molecular karyotype, well, that seems genuinely transformative for clinical diagnostics. And this is precisely where the OMCAR method, detailed by Reyes DeCordy and colleagues, really shines, doesn't it?
Absolutely. If you connect this to the broader diagnostic landscape, traditional karyotyping, G-banding, it's foundational, of course, but it's limited by its relatively low resolution. And it relies heavily on significant manual expertise for interpretation. OMCAR tackles this head-on by computationally generating a virtual karyotype directly from the OGM data. It effectively bridges that resolution gap between conventional cytogenetics and high-res sequencing.
Can you walk us through OMCAR's core purpose and maybe briefly how it processes the OGM data to create this virtual karyotype?
Sure, its main goal is to identify large DNA lesions, aneuploidies, translocations, large deletions, duplications, and generate that comprehensive virtual karyotype. Really crucial for constitutional disorders. The process essentially takes the structural variant and copy number variant calls from the OGM analysis as input. It then encodes these into something called a breakpoint graph. Then, it uses some sophisticated math integer linear programming to recompute copy numbers, ensuring everything balances out genomically. And finally, it identifies constrained Eulerian paths within that graph, which represent the actual chromosomes of the individual. It's a robust algorithm designed to reconstruct complex karyotypes accurately.
Sounds computationally intensive but powerful. How is its performance validated? Particularly thinking about diagnostic reliability here.
They did extensive validation. First, using simulations, 38 whole genome simulations of known constitutional disorders. OMCAR reconstructed the karyotypes with really impressive metrics. 88% precision, 95% recall for SV concordance, and a 95% Jacquard score for copy number concordance. These numbers signify a high degree of diagnostic certainty, which is obviously crucial for any clinical application. And then, maybe more importantly, they applied it to 154 real clinical samples, prenatal, postnatal, parental samples. It reconstructed the correct karyotypes in 144 out of the 154 cases. It achieved 100% detection for aneuploidies, 25 out of 25, and for balanced translocations, 32 out of 32. And it accurately called 72 out of 82 unbalanced variations. So very strong real-world performance.
That's a significant clinical impact, especially for those complex or subtle rearrangements. Did OMCAR enable any novel discoveries, cases that other technologies had maybe missed?
Absolutely. It identified plausible genetic mechanisms for 5 cases of constitutional disorders that had remained unsolved despite other testing. These included conditions like Critty-Scott, Wolff-Hirschhorn, Down, and Turner syndromes. Cases often caused by large-scale rearrangements or aneuploidies that can sometimes be tricky to fully characterize with lower-resolution methods alone.
Could you give us maybe a couple of specific examples of how OMMCAR made a difference in complex cases?
Certainly. Take balanced translocations. OMMCAR successfully captures these rearrangements. Now, these are often missed by CMA or even exome sequencing because they don't change the overall amount of DNA. Yet, they're incredibly important for assessing fertility issues or inherited disease risk in families. OMMCAR visualizes them clearly. Another strength is handling inferred variants. Sometimes the OGM data might have a missing call or a conflict. OMMCAR's algorithm can computationally infer missing SVs by ensuring the breakpoint graph makes sense, genomically making it Eulerian. They showed an example of prenatal sample with a complex interchromosomal duplicated insertion. OMMCAR correctly inferred a previously missing interchromosomal SV call, essentially completing the picture even from slightly incomplete data.
And its ability to provide genetic explanations for previously undiagnosed cases, those diagnostic odysseys.
Yes, that's a major contribution. OMMCAR provided critical genotype to phenotype, or G2P, explanations for some previously undiagnosed postnatal cases. It did this by identifying disrupted neurodevelopmental genes. For instance, finding a PX8 deletion, or a WDFY3HNR and PD deletion, or complex translocations interrupting key genes like TNC2, MBD5, and SOX5. This ability to precisely reveal gene disruptions, even within complex balanced events that are hard to detect otherwise, is a huge step forward for resolving those difficult cases.
Okay, let's pivot now to a specific and highly critical application area. Acute leukemia. This seems to be where we really see OGM's power in deciphering those incredibly complex genomic rearrangements that are often just intractable with conventional cytogenetic methods. The work by Tsai et al. Highlights this well.
Right. Leukemia is notorious for its complex structural variants. Many are recurrent, prognostically significant, yet frequently missed by traditional tools simply due to limited resolution. OGM is proving itself as a single, powerful test that can reveal these critical alterations, and that provides essential information to guide diagnosis, prognosis, and ultimately tailored treatment strategies for these patients.
What were some of the key findings when they compared leukemia samples to healthy controls using OGM?
Well, as you might expect, the leukemia samples showed significantly more SVs compared to normal blood samples. On average, something like 477 insertions, 457 deletions, 32 inversions per leukemia sample. And intertranslocation swaps between different chromosomes, often highly pathogenic, were observed only in the leukemia samples, not in the controls. Crucially, OGM detected all the previously known SVs in the leukemia samples they tested, with just one exception. And importantly, it found many additional novel SVs as well.
Can you highlight maybe a few specific complex leukemia cases where OGM provided really unprecedented insight?
Sure, in case one, this was a patient with B-cell acute lymphoblastic leukemia, BALL. OGM revealed an incredibly complex sequential translocation involving 4 different chromosomes, 5, 8, 12, and 21. A real mess, cytogenetically. It also precisely identified the clinically significant ETV6.RUNX1 and BCTT1.BALC gene fusions, and it detected a deletion on chromosome 12 that had been completely missed by conventional karyotyping.
Wow, another case.
Okay, case 2, also BAL. Here, OGM identified a huge deletion on chromosome 9p over 16 megabase pairs long, encompassing critical tumor suppressor genes like CDKN2ADB and also PAS5. And it also found a much smaller 88.5 kilobase pair deletion on chromosome 7 involving IKZF1. Again, providing a far more comprehensive genomic picture than they got from MLPA and standard karyotyping combined.
What about a more challenging gene fusion detection? Sometimes those can be tricky.
Yes, case 3, another BAL patient. OGM detected a terminal translocation between chromosome 6p and 14q. And critically, it detected an IGH.DSP22 fusion. Now, this fusion is more typically associated with chronic myeloid leukemia or lymphoma, and the DFA22 gene is located in a region that's often difficult to resolve and identify by conventional karyotyping, just due to its genomic location and complexity. OGM nailed it.
So OGM detects these large-scale SVs and their locations with remarkable accuracy. But if you need the exact breakpoint sequences down to the single base pair, is that where synergy with other technologies comes in?
Precisely. That's where OGM and whole genome sequencing, WGS, really achieve powerful synergy. OGM is brilliant at detecting the large SVs and pinpointing their genomic locations, the overall architecture. Then, WGS can come in to define the exact breakpoint sequences, maybe down to a few kilobase pair resolution or even finer if needed. This combined approach is proving to be both fast and cost-effective. And importantly, it bypasses the need for cell culture, which is often required for traditional karyotyping and FIH. That's huge in acute clinical settings where getting rapid, comprehensive results is paramount.
Okay, so the utility of OGM clearly extends beyond just that initial discovery phase. It's also proving to be an indispensable follow-up tool, isn't it? Particularly when standard of care methods leave ambiguities. This is explored really well by Drumsek and colleagues.
Yes, and this raises a crucial question for clinical practice, right? How can we best leverage OGM to resolve the breakpoints of, say, complex duplications or balanced structural variants when our traditional methods, maybe CMA or karyotyping, fail to provide sufficient resolution for a clear clinical interpretation? Especially in challenging scenarios like prenatal diagnostics or maybe when family studies aren't feasible or aren't informative.
So what's the proposed clinical pipeline for using OGM in this kind of follow-up capacity?
The pipeline they propose is quite logical. It involves using OGM as a targeted follow-up test when initial standard of care methods, perhaps microarray, karyotyping, or even whole exome sequencing, detect potential pathogenic SVs but lack the necessary resolution for a precise clinical assessment. It's about getting to that definitive answer when the first-line tests leave questions.
Can you share some specific clinical cases from their study that really showcase OGM's utility in this diagnostic problem-solving context?
Certainly. They had several compelling examples. In case P1, this was a prenatal case. CMA had detected two X chromosomal duplications. OGM was used to figure out the structure. It revealed a complex rearrangement where the duplicated material was actually inserted downstream of the STS gene, but crucially, in a region that didn't disrupt any known genes. This detail allowed it to be classified as likely benign, absolutely vital information for parental counseling in that situation. They later confirmed the exact breakpoints with long-range PCR and Sanger sequencing, guided by the OGM map, showing how OGM can direct further investigation.
What about resolving something like a suspected inversion, where conventional methods might struggle with the breakpoints?
Yes, case B2, another prenatal situation. Karyotyping suspected a paracentric inversion on chromosome 7. OGM confirmed the inversion, but critically, it precisely located the breakpoints within large, homologous segmental duplications. Knowing the breakpoints fell within these non-coding duplicated regions allowed clinicians to confidently rule out the disruption of known genes. This led to classifying it as likely benign, a finding later confirmed by SH. Again, that precision was key for accurate risk assessment.
And what about using it to investigate cryptic SVs maybe suspected after whole exome sequencing results?
Right, case B3 was an interesting postnatal follow-up. WES had shown zero coverage for exon 13 of the SENN1B gene, raising suspicion of a hidden SV linked to pseudohypoaldosteronism. OGM detected a homozygous paracentric inversion on chromosome 16, and crucially, it showed the distal breakpoint definitively overlapped SCN1B exon 13, confirming the gene disruption mechanism. Subsequent long-read sequencing determined the exact breakpoint, but OGM provided the crucial structural context. And beyond just the diagnosis, the OGM data also facilitated the development of a custom PCR test for carrier screening within the family, directly impacting clinical management.
So in these cases, OGM really was the missing piece of the puzzle. It provided that necessary resolution to move from suspicion or ambiguity to a much more definitive diagnosis or classification.
Precisely. These cases powerfully demonstrate that OGM provides critical, often otherwise unattainable, information for clinical assessment, especially in those tough prenatal settings or when informative family studies just aren't possible. Ultimately, this enables clear, actionable reports, significantly reducing diagnostic uncertainty and, importantly, the associated patient distress by providing a definitive answer much faster.
Well, what's truly evident here, I think, is how OGM, especially when you combine it with advancements like multicolor mapping and these automated interpretation tools like OMCAR, well, it isn't just an incremental improvement anymore. It seems to be fundamentally transforming our ability to visualize, understand, and interpret structural variation right across the entire human genome.
Indeed, the compelling examples we've walked through today really highlight OGM's remarkable capacity. It can resolve complex, previously intractable genomic puzzles, everything from pinpointing precise breakpoints in rare inherited disorders to untangling those intricate, prognostically important rearrangements we often see in acute leukemias. It undeniably complements and significantly extends the capabilities of our existing diagnostic toolkit, paving the way for more comprehensive and ultimately more accurate genetic diagnoses in clinical practice.
So, what does this all mean for you, our listener? Whether you're a clinician navigating complex patient cases day to day, or perhaps a genetics specialist in training, think about this. The increasing integration of OGM into clinical pipelines allows for a level of genomic insight that frankly was once unimaginable. As this technology continues to evolve and become more accessible, consider how might this ability to achieve such detailed whole genome structural mapping quickly and cost effectively, how might it reshape our fundamental understanding of disease mechanisms and truly revolutionize personalized treatment strategies in genetic medicine in the coming years?
It's an exciting time. There's always more to learn in genomics, and OGM, I believe, is just beginning to show its full potential for clinical impact.